First of all, I'd like to thank the organizers for giving me this opportunity to present our recent work on phase separations involving disordered proteins. I'll be mainly talking about the amino acid sequence dependence. So basically, when you replace one particular amino acid with another amino acid, what will be their effect on the phase separation propensity? And then I'll also show that how we develop the methods, to calculate the phase diagram of intrinsically disordered protein. So this will be the two prime focus of my talk. Compartmentalization is an important feature of biology. Like if you look the cell, it has a lipid bilayer membrane around it, so that keeps it distinct shape. And also, if you look at the organelles inside the cell, they also have this lipid bilayer membrane. So the intercellular space is divided into many small parts, but the cell can conduct hundreds and thousands of chemical reactions independently without any interference. However, recent experiments have shown that there are a lot of organelles which do not have this lipid bilayer membrane. They float like liquid droplets. They can fuse together, and these are mainly contents like proteins and RNAs. And physiologically, they are important. They carry several important biological activities, and some of them can actually, form some sort of aggregate, as my previous speaker also discussed, associated with neurodegenerative diseases. So this is a kind of old list of the examples of biomolecular condensates which have been identified using different experiments. And if you look at this list, most of them are actually inside the nucleus. But this list is not complete. In fact, it is increasing very rapidly. Experiments also suggest that intrinsically disordered proteins play a very important role in phase separations. So these other proteins are mainly like they don't have any unique folded structure and contain a lot of polar and charged amino acids. And also they have these low complexity sequences which my previous speaker elaborated in detail. So I'll not go in details here. But basically they contain some amino acids which, like if you look at the sequence, this amino acid comes often and often. Okay. And also these disordered proteins, they offer flexibility and can induce weak multivalent dynamic interaction. I'll come to that later. So if you look at in general phase diagram, what happens if the protein concentration is above certain limit, we call it some threshold limit, then the protein starts to form droplets. But this concentration is not unique. Like you can tune the concentration by changing the temperature, or experimentalists prefer to add some salts or change the pH, something like this. So you can actually change these things. And they usually observe these drop under the microscope when you add some sort of fluorophores like GFP or TAPI and all those things. And they actually look very beautiful, under the microscope. Okay, so now let us understand this typical liquid-liquid phase separation, diagram. So what I have here is it's not a real phase diagram; it's for general understanding. So on the x-axis, I have some concentration of the protein, and on the y-axis, I have certain parameters. That parameter can be temperature, it can be some ionic strength coming from salts, or it can be pH. Now within this blue region, the system undergoes phase separation. So if the concentration is within this limit, then it would phase separate and it would produce two distinct phases. One is the dilute phase, another one is the condensed phase. Now, if you go along this parallel dotted line, I have four different regions: 1, 2, 3, and 4. So this region 1, it's a one phase system. It's out of that blue region. And you can see here in region 1, I have molecules scattered here and there. The interaction is not very high. The moment you enter into point 2, it starts forming droplets. So you have a lot of small droplets forming. If you go even more at point 3, now you have a big droplet. Okay, it's a more condensed phase. Now, once you cross this phase diagram and come to some point 4, it's a solid aggregate. It's not a phase separate state. So once the concentration only within this blue region, the system will phase separate into two parts. So these are the typical interactions that govern the LLPS. Like disordered proteins contain a lot of charged amino acid residues. So electrostatic interaction is very important here. And along with it, you have a lot of dipole interactions. We have aromatic interactions in the form of cation pi and anion pi and all these things. And if you look at the low complexity sequences, which I talked about, like they have a lot of arginines. So what this arginines is doing, arginine is positively charged, so it can give you electrostatic interaction. Also, arginine has some sort of a double bond like this, so it can give you little bit pi interaction. So one amino acid can give you multiple type of interactions. Similarly, if you look at tyrosine, tryptophan, phenylalanine, they have like pyrite structure, and they can also give like cation pi interactions, pi pi interactions, all these things. So one single amino acid is giving you multiple type of interactions, and all these are happening in the dynamic environment of the biomolecular condensates. Now, if you look at the energy scale of these amino acids, it's not very high; it's a weak. So typically we talk about weak dynamic multivalent interactions in the context of LLPS. Like if you follow the literature, you'll find this term many times. And this actually is governed by these amino acids. Similarly, RNA and ATP, they can also influence the phase separation propensity. So what happens in case of RNA? RNA is negatively charged. So it has a lot of phosphate group. So it can give electrostatic. And along with it, you have a lot of this pi-like structure. They can give like, aromatic cation pi and pi pi type of interactions. So that's why there's a lot of literature study there that shows that RNA can critically influence the phase separation propensity of proteins. And there are a lot of literature study that actually shows how important is this multivalency. I'll not go details here. And also, like it can lead to diseases. For example, this fast protein, or talk about some other proteins, human tau proteins. They also can form, solid-like aggregates and associated with various neurodegenerative diseases like ALS and all these things. So there's a lot of interest going on there. How this coming, like how this aggregate is forming. So yeah. So when we started, doing simulations, like starting the phase separation is a bit tricky because it's a collective phenomena. So you need a lot of protein molecules, and that too with disordered proteins. So so you have a very big colossal system, and you cannot really go for an all-atom simulation. So you have to stick to coarse-grained model. And at that time, we didn't have any, definite method how to study a phase separation of these big size. So what we did here, we started with the lattice model. It's a fairly simple model, well studied, and we had this sorry, these two proteins, SV1 and SV15. These are not real proteins. These are kind of model proteins, and has been studied extensively in the context of LLPS. Now, if you look at the sequence here, you can follow the color red, blue, like this. So this one is basically EK. The red one is the E amino acid, and the blue one is the K, lysine. So if you follow the charges, so you have basically -1, +1, -1, +1 like this. So the first sequence, that SV1, it's a strictly alternative charge pattern you have. And for SV15, you have certain blocky charge. And then our potential function was fairly simple. We only had like excluded volume and electrostatic interactions. And if we follow the electrostatic interaction, SV15 will have more attractive interaction. So it will be more prone to phase separate. This is exactly what happens here. Like if you look at for SV15, like it has a higher critical temperature. And we also have certain snapshots associated with the dilute phase and condensed phase. You can see in the condensed phase, they are more, a lot of molecules are there, more compact, and in the dilute phase, they are more scattered here and there. So far, so good. But when we move to more, charged pattern sequences, for example, you can consider die block. So die block, basically, you have one big positive charge block and one big negative charge block. But we found that the system is somewhere stuck. It's not moving at all. It's not doing anything. So we had to move from lattice model to off lattice model. And this is called the slab simulation methodology that we and few other groups are using for a very long time. So what we are doing here, we first energy minimize the system, remove all the bad contacts, and then we do simple NVT at high temperature. Okay. And then we compress the box at high temperature. So that actually produces the condensed phase. Remember, the condensed phase, the protein has a lot of density, very high density, right? So here we are producing that. And after this NPT, we are essentially expanding the box along one of the axes, let's say Z axis. And that actually creates this slab. Now, we are equilibrating it, this slab, at different temperatures and calculating the density or number density along this Z axis. And this actually looks like this at different temperature. So when your temperature is the lower, this peak height is higher. When you have a high temperature, the peak height is lower. Okay. So this will be like, if you follow this arrow, this will be the density on the condensed side at different temperature. And this will be my density on the dilute side, right? So if you now connect those points as a function of temperature, you will get both the dilute side and condensed side. And then we use some sort of approximate scaling relations to get the critical point. So another alternative way could be like you take a very big box and then you put all the molecules there, do simple NPT, and after some time you find that you have a big droplet here. Now the problem with this particular method is it's like 3 to 5 times computationally more costly. So we don't consider this method and continue with the slab method. It's again a coarse-grained model. So essentially, we consider each amino acid as just a single residue. We took 100 molecules. Actually, we tried with 100, 200, 300, something like this. And we found that if you have like 100 molecules, the statistics is good enough. The potential function is again fairly simple. We have bond, we have electrostatic interactions, and then short range interactions. Typically, we go for like 2 microsecond equilibration and 4 microsecond production run. And we also have run for more than 6 microsecond, and we found okay, if we can continue up to 6, it can the statistics is trustable. Now, the interesting part, like the bond interaction and the electrostatic, these are fairly simple. The interesting part comes in the short range. So there are different ways that you can actually treat these short range interactions. Different groups have published different papers. So what we are doing here, we are using the cage potential, this scheme Hammer potential. This is based on the contact, okay, C alpha contacts. So now we have like 20 different amino acids in our simulations. So if you consider the pair, we have roughly 210 pairs. So we have 210 interaction energies there in the contact maps. And this is done long ago, like in 1996, the first paper came, the second paper came in 2008. So the idea of this contact map based energy has a very long and rich history. The interesting part here is that if you look at the most of the pairwise energy, these are mostly like standard LJ, Liardio's interaction. So this is attractive in nature, and some of the interactions are repulsive. Like it's all positive. This is my zero axis. Like most of them are negative, and some of them are positive. So what this particular scheme does, that it actually differentiates between two type of energies. So if you have some pairs or most of the pairs, they are attractive in nature, and some other pair they are repulsive in nature. For example, let's say arginine, lysine, arginine, aspartic acid. So this kind of interaction are mostly repulsive in nature. On the other hand, if you consider like tryptophan, tyrosine, phenylalanine, all those aromatic things, they are attractive in nature. Okay, now actually looking at the simple simulation snapshot, you can actually differentiate whether the system is phase separating or not. So if you have a phase separated state, you can see a lot of molecules there sticking at the center, and when it's not phase separated, the molecules are scattered. So just by watching at the simulation snapshot after your production run, you can actually clearly see whether the system is undergoing phase separation or not. Now, so far so good. We talk about this IDP, and IDP has a lot of charged residue. So the electrostatic interaction, they play a very important role here in LLPS. Now typically we use this equation to calculate the electrostatic interaction, and one of the factor here is the dielectric constant, and typically we choose a fixed dielectric constant. But in phase separation, what is happening, like you have a dilute phase and you have a condensed phase. The dilute phase basically contains more water and less protein, and the condensed phase is you have more protein and less water. Water dielectric constant is around 78.4, and for protein it's very less. If you consider a fully folded protein, it's around like 2 to 4 maybe, and for disordered protein it may be a little bit higher, but the difference is large. So when you have a particular molecule in the dilute phase, the dielectric environment that it's filling is very different than the environment it's filling in the condensed phase. So that molecule, when going from the dilute to condensed phase, it will have a completely different electrostatic environment. So what we essentially need is a position dependent dielectric constant, right? But that is difficult to implement in the whole code. So what we did, we use different values of dielectric constant. So it's not very difficult to calculate the dielectric constant. You can actually do from time dependent dipole moment fluctuation, and it would look something like this. We calculated this thing for DDX4 using Amber force field, and we actually use a lot of water potential to check how much is the fluctuating. So you can actually take some values around this and consider this as the dielectric constant of the whole system. And then when you plot it as a function of the protein, you can see there is a decrease. There's a decrease. Like if you increase the protein concentration, the whole dielectric constant of the system is decreasing, right? And there are also certain experimental papers. This is from dielectric relaxation paper, they actually did some dielectric relaxation experiment and showed that inside the cell it can be very different. It actually depends from species to species what will be the dielectric constant. But important thing here, it's not water like. This is less than 78.4. And when we applied this thing, different dielectric constant, we found around a value of like 40. The critical temperatures that we are getting for 4 different variants of DDX4 that matches, at least qualitatively with the experimental data. So we continue to use this dielectric constant of 40. Now, it's very important that we are dealing with liquid-liquid phase separation. So there must be some proof to show that it is indeed liquid. It's not like solid or it's not like gas. And there are two ways that you can actually show. One is by looking at the radial distribution function, because we know that solid, liquid and gas, they can have a completely different RDF. And also you can calculate the diffusion coefficient and show that, okay, the molecules are moving. So we actually calculated them and show that we have certain diffusion coefficient. We didn't compare with the experiment, we don't have it. But we can actually ensure that from this data that this is not something arrested state. The molecules are moving, it's dynamic. Now we concentrated on this caprin-1 protein. This is also an RNA binding protein. It also has a lot of RGG regions. And if you look at the sequence, there are a lot of RGG and RG, something like this. So it can be also very interesting from your perspective. See, we are not taking the whole protein here. We only are taking the disordered proteins. In fact, if you look most of the literature of the simulation in the context of LLPS, you will see that we are only mainly taking the IDP part, not like full length of the protein. So this wild type caprin-1 in the disordered region, it has like 103 residues. And out of this, we have 15 arginine, 1 lysine, and 3 aspartic acid. So 15 plus 1 plus 3 is total 19. So you have 19 residues out of 103 residues are charged. So this is a strong polyelectrolyte. So its phase separation is likely to be governed by like similar polyelectrolytes. And then what we did, so these red lines, these red dots are basically the position of the arginine residues in the whole sequence. Now what we did here, like if you look at these 3 variants, we are changing 4 arginine residues with lysines. We are only replacing 4 arginines. So for this first variant, these first 4 arginine residues are replaced by lysine. For this middle variant, we are replacing these 4 arginines by lysine. And for this C variant, we are replacing these 4. Okay. So if you consider these 3 variants, we have the same number of arginines and same number of lysine, but their position is different. The question is whether we can see some difference in their phase separation propensity or not. Now, in addition to that, we also have this 15R to 15K residue. So basically here we are replacing all the 15 arginine residues with the lysine. And then in addition to that, we have this phosphorylated caprin-1. Okay. So basically the experimentalists, what they do, they can synthesize this thing or probably chemically modify and add this phosphorylation group. But in our case, because it's like C alpha based model, it's kind of a tricky. So what we did, we considered the tyrosine residues and added -2 for simplification. Okay. And also there is mass spectroscopy data that shows that the number of phosphorylation group that has dependence. Like it's sometimes 6, it's sometimes 7. So you have a distribution. But that is again very tricky to implement in the simulations. So we considered all the 7s tyrosine residue and added -2. So if you consider the charges for this phosphorylated version, now the charge is -1. So it's a polyampholite. Okay. Now your wild type caprin-1 is polyelectrolyte and this PY caprin-1 is a polyampholite. Now we are asking what will be the effect on their phase separation propensity of all of this when you add the salt. When you are adding the electrolyte, what will be the change in the phase separation propensity? So this is my phase diagram. So this is for the caprin-1 at different salt concentration, and this one is for the PY caprin-1 at different salt concentration. So we consider 4 different salts here, 0 millimolar. So basically there is no salt except few ions you have to add to neutralize the whole system. And then we have 200, 480 and 960. 3 different salt concentrations here. Now if you consider this one, this is PY caprin-1, it's a polyampholite. So at 0 salt concentration, you have the highest phase separation propensity. If you look at the peak value, this is highest. This y-axis is the temperature axis. Now, as you continue to increase the salt concentration, this value is going down from 0 to 200 to 480 to 960. So, what is happening here, when you are adding a lot of salts, the electrostatic interaction is screened by the salts. So, that's why the phase separation propensity goes down. Now, if you look at here for caprin 1, it's a polyelectrolyte. So here at 0 millimolar, this red one, it has the lowest phase separation propensity if you look at the temperature scale. Now, when you are adding salt, this goes down, goes up. You can see the 200 here, this one, orange one, this is 200 millimolar salt concentration. If you add further salt, let's say go to 480 millimolar, it's increasing again slightly. And if you increase to 960 millimolar, then it goes down. Okay. So what is happening here? As you continue to increase the salt concentration, the phase separation propensity first goes up. If you are following the gray line, it goes up and then it goes down. So you have some sort of re-entrance phase here. So what is happening exactly here, that if this caprin 1 is positively charged, right? So you have a lot of repulsion there. You are adding salts. So that repulsion is screened. So the phase separation propensity that goes up. At an optimal concentration of, let's say, 480 millimolar, it becomes even higher. Now, as you further increase the salt concentration, it goes down because you have now more and more screening. Okay. So that's why we have this re-entrance behavior. And then we also did this for different variants. So on my left hand side, I have 15R to 15K. So here we replace all the arginines with the lysine residues. Now here I have three different other variants, N, M and C. Now if you look at in general, like this one, it has the lowest phase separation propensity across the all salt concentration, because arginine can provide more interaction to the system compared to lysine. Although they have the same charges, but the latest interaction for arginine is higher than lysine. And this is well documented in the literature. Now here, for the position dependent, you see more of these two, this N and C, they are more or less similar. But here, in case of the middle, this is a little bit higher. So what is happening when these things are very close to each other? There is little bit repulsion there, which is not here in the M variant. So that's why this M variant has a higher phase separation propensity. Now we are interested in how is the salt distribution, like what the salt is actually doing for caprin 1 and PY caprin 1. So you can focus on this snapshots here. So what essentially we have, this data is for caprin 1 and this data is for the phosphorylated caprin 1. And we are showing the distribution of Na and Cl at 480 millimolar. So this is caprin 1 number density distribution, chloride and sodium. Now here we are showing the same thing, but without this caprin 1, but the unit it 5 minutes. But the unit is different. It's in millimolar unit, and you can actually see when the caprin 1 density is very high, even it's a positively charged thing, you can actually take a lot of negatively charged chloride inside it to balance the charges. And at the same region, see the positively charged sodium are repelled a bit. Now here, in case of the phosphorylated one, the charge here is -1. So it's taking little bit of sodium, and at the same time, some chloride is also coming because there are a lot of positive charges there. So both are coming here, but for the sodium, it's higher because overall the net charge is negative. So this is well expected. And we show some of the snapshots here. This is my full box snapshot with all the salts and all the protein molecules. Now, if you remove the salts here, you can see this is my condensed phase, and these are dilute phase, like very few molecules are there. And if you remove the sodium, it's like a more or less homogeneous distribution. And if you can see here, maybe not visible properly, you can actually see there is a little bit more chloride ions here. So what is the role of that chloride ions? Now, when we look at the snapshots of this condensed phase, this thing here, we actually see there are some bridging interactions here. So what is the role of the chloride ion? Your caprin 1 is positively charged. When you add the salt, the electrostatic interaction is neutralized, right? Because it's positive, chloride is negative, so it can neutralize. But is it only neutralizing it, or is it also acting as a bridging agent? Remember, if it is acting as a bridging agent, then your apparent repulsive interaction can be tuned into a attractive interaction. So you have two arginine, two positive charge repelling each other, but now in between you have a chloride. So this repulsion between these two arginine is now becoming attractive in nature. So indeed, if it is playing as role as like bridging agent, then the phase separation probability will go up. Okay. So this is typically like radial distribution function here, and you can see a lot of interaction is here, and we can actually see that 98.5%, almost all of them are actually within this fast peak minima of the arginine. And then we asked like whether you can define in a definitive way whether the chloride ions are indeed acting as a neutralizing agent or bridging agent. So what we did here, we actually calculated the energy of this trio. You have arginine 1, arginine 2, and in between you have chloride. So we calculated the interaction energy involving these two arginine and one chloride. And if the interaction energy of this trio is negative, so then chloride is providing you attractive interaction. So now it is acting as a bridging agent. If the interaction is positive, then it's only neutralizing it. It's not acting as a bridging agent. And then you can have some sort of intermediate. What we found something very interesting, that almost 87% of the chloride ions, they are acting as a bridging agent. So that is actually promoting the phase separation here. Sorry. Yeah, very quickly I can go some of the limitations. So far so good, but this methods and all those things that I have shown has certain limitations. For example, it's a Corscan model, obviously, so we are missing a lot of detailed interactions. The pi interaction, cation pi interaction, all these are super important as shown by many experiments. The way we are treating it's not like explicit. And then when we are dealing with the simulations, we usually don't take the folded domain. Sometimes you can treat it as some sort of inert body, but mostly we discourage them because integrating the force field with folded and disorder is kind of a bit tricky. It's somewhat like gray area. So, but there are certain experiments that actually show that this can be super important. For example, in case of FIB1, these are RNP, ribonucleic protein, they actually show that these folded domains can be very important in phase separation. So that has to be taken into account. Now, in simulation, the methodology that we use that actually shows only one droplet, but you can see a lot of experiments are there. They can show multiple droplets are forming, but we cannot take into this account. And then role of water molecules. We know in biological system water are very important. We cannot take them because the system size will be very high. Force field parameter for IDPs. As you can see, like some of the result that I have shown, the distinction is not very high from the experimental result. So we need to tune those force field parameters in order to be more accurate with the biological thing. The last one, conclusion. Okay, so it's LLPS is fundamentally important and has a lot of involvement in biological activities. You have a complex set of interaction that is operating that actually tunes the phase separation propensity. And yeah, we stop it here. And thank you everyone for listening. I also have to thank Professor Yusen Chan, University of Toronto. We have a long collaboration going on. Professor Julie Foreman K, Louis K. They have a lot of NMR data for LLPS, and I have been super benefited in discussing all the experiments. I actually learned how to read and experiment papers from them. So yeah, finally, thank you everyone, and I'm happy to take few questions.
Yeah. So before you go on, I have one question. So this slab geometry that you are talking about, how do you ensure that it's equilibrated? Because it's just doing it arbitrarily. It seems to me completely arbitrary.
So when you say arbitrary, you basically means like how much we should expand, right?
No. So when you say okay, you're just kind of compressing it and then
Right.
expanding it.
Right. So the idea of compression is that we have to create a condensed phase, and when compressing the box, I'm getting the condensed phase. Now I also need the dilute phase. And how do you
Well, but it's not just that, right? I do not know a priori what is my free energy minimum. So it can have different kinds of structures that can emerge. You're just forcing it to form a
Right.
condensate and you're
So,
then doing it.
yeah, I
Is there a
understand.
mathematical like a proper physical argument?
No,
to
I have
that?
to say that we are focusing. It's more like you can actually get it quickly. Otherwise, it's very hard to deal with these kind of things.
I
Because
understand that you're doing it, but it seems completely arbitrary to me. I'm not familiar with this literature.
so we have like not only us, but there are also other groups. They have checked a lot of things. For example, how much you should expand. Okay, and all these things. What should be the initial concentration? It really does not fluctuate a lot.
But it's not just see, you can do the simulations, but at the end of the day, you're like doing experiments. But in terms of the actual thermodynamics and our own understanding of StatMech, how do you ensure that the system is equilibrated? There is no other way, right? I mean, you have to, that's not.
I mean, like standard MD, you also check all those energy values to ensure that it's kind of the fluctuations are within the limit.
I see. Okay. Okay. This we can
And there
discuss
are
later
also theoretical papers that have actually checked that the surface effect is also minimum when you have a slab, compared to a full box.
I see. I see. Okay, let's
But
discuss.
I agree there are certain limitations with these methods. But as of now, in order to deal with this huge system, we don't have any better methods available. Yeah, I mean theoreticians have always this thing. I agree. Yeah, sure.
Hi, great talk.
Thank you.
I'm just thinking that the finite size effect will have a very difficulty here, right? Because you are just taking a single condensate
Right.
and you really don't know what would be the real size of that because it is limited by your number of molecules you are taking.
Right. Actually, we checked up to like 500, 600 protein molecules, and we didn't find any difference in the phase diagram.
Okay, so you mean
Right.
it doesn't depend on the size of the
No, it does not depend until and unless if it is if you're taking 20, 30, there are some papers available. There, the system size dependence can be important, but we have checked.
Okay. Another thing is that you discussed about dielectric constant, right?
Right.
You said you have used blanket 40 for all.
Right.
So within the condensate as well as outside.
Right.
But that is too mean field.
Yeah, that is also an approximation. I agree. That is also an approximation. So our point was that we're not going to use something very low and something very high.
No, I understand, but you have to understand that you are making it too mean field, right? It is not even dependent on the concentration of the proteins in the condensate.
True.
It is in dilute.
True. But then if you want to do it in a real way, then it would be, it would be nearly impossible. That how do you implement it? It also varies from
you can still have a protein some local concentration dependent dielectric constant.
Yeah, something like this can be implemented. True.
That will be much better, I guess.
Whether that will be better or not, I don't.
And just last one comment I have is that because you are working in a C alpha model, if I understand correctly.
We only take C alpha.
So, see, thinking about our sandwich chloride atom between two arginine and then thinking about arginine as a C alpha atom, it's like essentially C alpha atom interacting with another C alpha atom, which is a chloride atom.
Right.
And that is very different from two arginine interacting.
Yes. Right. But within that force plane regime, this is only what I can do.
I completely understand. I'm just thinking that how these effects will play out ultimately in the real scenario.
In the real scenario, then we have to depend on the experimentalist to see how it happens.
Okay.
But I think there was some other papers also, something like this.
No, I'm not contesting. I know there are other papers.
The whole field is basically like C alpha based. So as of now, maybe Martini type of force field may work, or if you go from C alpha to
because this also doesn't have the excluded volume effect currently, right? Because you're just compressing everything.
Yeah. So that's why we are claiming that all the results are kind of qualitative. We are not going into the quantitative.
Suman, I was looking at some numbers and you had 900 Kelvin on your slides in the initial.
Yeah,
as an experimental biologist, I get worried about this.
Things are for lattice models, and you can actually have a lot of high temperature. That's not not physiological. So what we are interested whether it gives higher critical temperature or not. That's it. We're not looking at the
But at physiological temperature, would that still hold true? That was my query.
In physiological, we're getting around 350, 360 for critical temperature, something like this. But if you want to do it under physiological temperature, then we have to vary salt concentration or pH. And again, those things are very tricky, to control in simulations.
Okay, I can talk to you more in the break. Thank you.
Yeah, actually, this is reduced temperature.
Hi, very nice talk. So I'd like to ask that all the phase diagram you have shown, so there is a short arm and there is a long arm in
Right.
all of them. So
Right. This is again a limitation of the method. If you look at the dilute side, it's not bending too much. It's only the condensed part that is bending. This is again a limitation.
okay.
We have a lot of limitations of this method. But as of now, if you want to deal with this kind of system, this is the only thing available.
Another observation, I might be wrong, but I feel that those phase diagram which has a lower UCST, it looks more broader than the.
Right. It's because of the fitting function that we are using. It's kind of an arbitrary. So because of that, it's actually in the critical temperature, it should be like single point, right? But it's kind of comes flat.
Okay, thank you.